Hello, this is Dr. Huda. I hold a PhD in microbiology from the Faculty of Sciences and Techniques in Tangier, Morocco. I am also a teacher in a health institute and a part-time teacher at the University of Tangier. Today I'm pleased to present my research project under the title of Epidemiological and Molecular Study of Fluoroquinolones and carbapanems resistance Enterobacteriaceae involved in community infections in northwest of Morocco. Enterobacteriaceae represent a heterogeneous family of bacilli gram-negatives, which include a large number of genera and species. These germs can be commensals of the digestive tract in humans and animals, as they naturally exist in the soil. Further, these Enterobacteriaceae are common pathogens causing different types of hospital and community-acquired infections. All bacterial infections are treated with antibiotics, which act on different levels in bacteria to ensure its destruction or block its growth, such as inhibition of the cell membrane, the DNA, or protein synthesis. These resistant genes are carried by chromosomes, therefore they are transmissible between species of the same family, or they are carried by plasmids transmissible between different species and bacterial families, which promotes a large spread of resistance. Fluoroquinolones are currently used to treat a variety of human infections caused by different bacterial families. It works by blocking the synthesis of DNA by its fixation on topoisomerase, leading to the death of the bacteria. However, resistance to fluoroquinolones can be chromosomal by modification of GRAS and topoisomerase, or plasmidic, called PMQR, carrying genes which either protect GRAS, like QNR A, B, C, D, and S, hydrolyze fluoroquinolone like AAC genes, or reject the antibiotic by the expression of efflux pumps. On the other hand, carbapanems have traditionally been considered the drugs of choice for infections caused by Enterobacteria producing beta-lactamase enzymes because they are not affected by this resistance mechanism. So, the massive use of antibiotics among humans and animals, the self-medication, the no-respect of the antibiotic dose, all these lead to the evolution of bacteria that become multi-resistant. Generally, the emergence of multi-drug resistant strain in the hospital environment can be controlled by the screening of patients at risk and the control of internal hygiene, but in the community, controlling the spread of MDR is particularly difficult. In recent decades, therapy of invasive infections due to MDR is challenging worldwide, especially the emergence of PMQR and CPC plasmid transferred both within Enterobacteriaceae into other bacterial families. In Morocco, several studies have been carried out on the evolution of resistance of Enterobacteria to antibiotics, particularly in hospital area. However, in northern Morocco, few data are published on this subject. Our main target is to study the spread of PMQR, CPE, and multi-drug resistant Enterobacteriaceae to antibiotics isolated from community patients in northwestern Morocco. This region is very known for its traffic and marine activities between Europe, and Africa. From different private laboratories, a total of 634 samples derived from several origins were subject to this study. The identification of collected species was performed by phenotypic and biochemical tests. In parallel, the evolution of susceptibility of Enterobacteria to 20 antibiotics was examined by phenotypic tests, and the search for broad-spectrum beta-lactamase and carbapenem resistant Enterobacteria is implemented by various phenotypic tests. In the molecular part, we proceed by the extraction of total DNA from each bacteria by thermal shock and plasmid DNA by extraction kit. The detection of carbapenem and fluoroquinolone resistant plasmid genes was performed by multiplex and simplex PCR. And finally, the confirmation of plasmid location of these genes is carried out by PCR on plasmid DNA. And finally, the confirmation of the variant genes is carried out by sequencing and RFLP analysis. From the total of 634 samples, we selected 577 Enterobacteriaceae, which represent 91% of Enterobacteriaceae, and included different bacteria species predominated by E. coli. This is a normal rate since Enterobacteriaceae are among strains most frequently isolated. In general, more than 80% of bacteria were resistant to penicillins, and more than 30% were resistant to the majority of antibiotics tested. Only amikacin and colistin and meropenem, which were active against more than 90% of the strains. In parallel, we detected more than 50% of multidrug resistant strains in all identified species with high rates. This epidemiological result obtained show a spread of MDR in community patients of northwest of Morocco. According to the estimated by Review on Antimicrobial Resistance, MDR will be the leading cause of death in the future, and statistics show the number of annual deaths caused by antimicrobial resistance is higher in Africa. This is probably due to several reasons such as poverty, incorrect use of antibiotics, and poor diagnosis of infections. Our data reveal a remarkable diffusion of the PMQR genes in different species of Enterobacteriaceae. The AAC gene was most present, followed by the QNRB variant. Klebsiella spp represent the most dangerous pathogen in this collection. It carries several PMQR and has important concentrations. Regarding the screening of CPE based on phenotypic tests, we have selected 6 strains capable of expressing a carbapenemase, among which 2 Klebsiella and 1 E. coli harbored CAPC and VM genes. At the national level, in the hospital environment, the emergence of CPE was published since 2009 in several cities of Morocco. These studies have shown a wide diffusion of OXA48 genes followed by EMP and NDN, unlike the VM and CAPC genes. However, in the community environment, very few data studies are performed. The CPE detected in this study are among the first community strains isolated in this region. So in this study we have determined a spread of multi-drug resistance Enterobacteria in northwest of Morocco in outpatients, detection of Enterobacteria resistant to fluoroquinolone with high MEC, an emergence of MDR carrying several big plasmids, detection of strains carrying 1 or more PMQR genes, detection of community enterobacteria producing ESBL and carbapanems, and finally detection of VM and KPC in community environments in this region. So this table represents the list of the most pathogenic Enterobacteria strains selected in this study, mainly E. coli and Klebsiella expressing several plasmid genes. And thank you for your attention.